And thank you all for showing up tonight. Before we get started, I just wanted to acknowledge that we are on the traditional lands of the first people here, the Duwamish people, both the past and the present, and I want to honor with gratitude the land itself that we are on and the Duwamish tribe. Let's keep them in our hearts when we think about these things. So we're here tonight for this event called Medical Angels, and it was one of those names that sort of struck me a while back when I was first forming the idea for this. And I'm so used to saying medical heroes. When we talk about people who participate in clinical trials, especially when they're doing something that's beyond themselves, they're not getting any sort of personal benefit. But it sort of made me realize the sort of legacy that there is around HIV and participating in trials, and the people who participated in those trials in the late 80s and early 90s who helped us to create antiretroviral therapy and get to the point today where some people who are newly infected could go on one pill a day and not have any sort of severe side effects. And it's all because of those people who were getting involved back then. And as I was putting this event together on Facebook, people would write little comments, and there was this one individual who just wrote this comment, and it just touched me. It was, my oldest brother was in a 20-year study in the early 80s at the College of Berkeley in California, and he's in heaven now, but because of his participation, he helped save lives, and I wanted to thank everyone involved in studies now. And I just thought, that sort of thing was really the spirit behind this. We're all medical angels in some ways, no matter how you get involved. And now that we're trying to cure HIV or create some sort of long-term durable control, it's going to take a bunch of angels. And so I guess I grew up in the 90s where angels in America was always in the back of my mind. And so medical angels is what we're calling today. This is going to be a conversation led by the community. So we have our three community people on the left side, and we have our three honored guests, and I'm going to let them introduce themselves to you all.
Hello, everyone. My name is Manuel. I am a Defeat HIV Community Advisory Board member. I'm a young person living with HIV. I've been living with HIV for the last 7 years.
Hello, everyone. My name is Tranisha Arzah. I've been living with HIV my entire life, 29 years now. And I'm also a Defeat HIV Community Advisory Board member. I formerly worked at a program called BABES Network, which is a peer-led, women-led program that provides services for women, including trans women who are living HIV and their families. And currently I work as an HIV STI tester at Gay City, which is Seattle's LGBTQ center. Thanks.
I'm Deontay. It sounds like Beyonce, but I sing better. I actually am a peer navigator for POCAN, which is known as the People of Color Against AIDS Network. It's been around for almost 30 years, advocating for people of color within our community. I'm also the NAACP's first LGBTQ chair. And within that space, I want to make sure that if we're going to talk about the advancement of our space, we have to talk about HIV when it comes to our LGBTQ community. And I'm really happy to be here as well.
About 25 years I've been working on cell and gene therapy with CAR T cells for HIV, and hopefully we'll get to talk more about that.
I currently live in San Francisco. I had HIV nearly my entire adult life. I'm a former ACT UP Golden Gate member. I've done quite a bit of activism and quite a bit of participation in community advisory boards. I was asked to be here because I was one of the people that participated in a gene editing study, and I've been off meds for 5.5 years with a viral load average of 100.
And no one asked him to. Hi, everybody. Matt Sharp. I'm from Berkeley, or I live in Berkeley, California right now. I'm originally from Amarillo, Texas. So, I usually don't say that. But it's great to be here. I always feel so comfortable coming back to Seattle, and I just want to thank everybody at Fred Hutch and Michael and everybody who's so gracious for me to come here every time that I ask and want to be here and join you and learn. That's what I do here, is learn a lot. So I was diagnosed in 1988, and I'm 63 years old. I'm a long-term survivor. I guess the reason I'm here tonight, and many of you may have heard or read about my story, is with the gene editing trial of Sangamo zinc finger. And I'm going to talk a little bit about some of the positive results, some of the really the results that weren't expected from this trial that I saw in my case and in many people's cases. I'll just finish by saying I follow really working now. My main focus of work is mobilizing long-term survivors and people that are aging with HIV across the country, and work with a project called the Reunion Project. And we were here in Seattle actually 2 years ago. Right now. Yeah.
Great, thank you.
Thanks.
Thank you guys very much. So I actually wanted to take a step before we start talking about the gene therapy. I wanted to actually go back to you guys as individuals. What was life like before the diagnosis? Who were you? Where were you at? Sorry, I feel like Oprah right now, so if you guys got to give me a question.
I was a little busy. Just saying, in the 80s, I'm one of those IV drug users that you think are never going to get sober, and ended up homeless in San Francisco. So I was a little busy. It was no surprise at all to me. Actually, I knew I was HIV positive when I lived in the state of Michigan, which has draconian reporting laws in the 80s, and there was absolutely nothing they offered you except for surveillance. You get calls from the health department all the time. It really sucked, and I left. So before before I learned that I had HIV, I was just a drug addict.
Just a drug addict. Thanks for asking that question. It is a question that doesn't get asked a lot, and I really appreciate it personally. But so I was a ballet dancer. I was in the middle of the very middle of a 25-year ballet career. Actually, that's a 15-year ballet career. I added 10 years to it, and I came down with, I got a test for HIV, and started watching my community die around me, like one at a time, and I fell into it. I just immediately said, I'm not gonna die from HIV. I'll get hit by a bus first, and maybe that's why I'm still here. Let's just make sure when we leave tonight that a bus doesn't come around the corner.
Oh, sorry, and Dr. Carl as well. Where were you at before this epidemic hit as well?
So, I actually trained here in Seattle in the 1980s in bone marrow transplant. And that was the first cure for HIV. I mean, for leukemia. And when you look at it, cancer, in some ways, when you get a diagnosis, it changes on a dime. And the same thing happens, like, what you just heard from the Mats. So the whole life changed. And so I started here because I wanted to work on the immune system. And at the time, in the early 1980s, the worst immunodeficiency was bone marrow transplant, because it took away your immune system with radiation and chemotherapy. Then AIDS happened, and I started working on that. And our first CAR T-cell trials started in 1996. We began, and a lot of people don't know that. They think, CAR T-cells are now marketed, for leukemia, but the first patients ever treated actually had HIV, and we did that then when there were no drugs. And it was just an awful carnage. And then, the drugs came out, and the industry didn't care about making something for HIV, and then it went to cancer. But so I have gone through this circle from working on HIV to cancer and HIV.
I also wanted to add that when I got my HIV diagnosis, it really felt like I got a whole disease of society with a virus. Like, you get all the stigma and everything else that goes with a virus. No matter who you are or what you do, it follows you. And that was like the most profound thing with, finding out that I had HIV.
So just to get a little more background before the Sycamore trial, have you all participated in any clinical research trials prior to this one? If so, what were some of those trials? What were the processes like? Were they similar processes to the one that we are wanting to dig more deeper into?
So Matt and I were talking about this on the plane ride over. Matt lives in San Francisco, I live in Berkeley, so we got to travel together. I wanted to say we know each other from, we were in ACT UP Golden Gate together and shared many strategies about how to get arrested and how to get attention. I have been in so many clinical trials that I lost count. Somebody tried to help me figure that out. I have it all filed away in boxes, my reports from some of the trials. I can't remember them all. But I was in some of the first AIDS clinical trial group studies. I was in the first Crixivan sequinavir combination study, or sequinavir combination study at Stanford, by the way. Anyway, millions of studies for pills. And then, because I started down that road, I developed multi-drug resistance as a result. As you all heard the story of sort of this recipe for disaster that a lot of us went down, which was, we were desperate to try anything we could get our hands on to save our lives. So we took the latest pill that came out, adding that to a virus that was already vulnerable and ready to escape around the new drug. And so eventually what happened is I developed multi-drug resistance and started looking for immune-based therapies. What a concept. Why don't we figure out something to change the body rather than the virus? And we developed a whole, Matt was a part of it with Martin Delaney at Project Inform, to push for immune-based therapies. And so one of the trials I was telling Matt about, that you talk more about your study experience, was I was involved in Dick Hong's thymus transplant study, which Carl knows about. I flew all the way across the country to Vermont to have a thymus transplant. And I'm not going to go into the detail. It doesn't sound as gory as it was, but it was very risky. I had a plant put in there of a child's thymus. These were babies that were born that had heart problems, and they had their thymus tissue removed at birth. And normally that tissue was just discarded. So Dick Hong was this brilliant researcher in Vermont who thought, well, maybe if we could take that thymus tissue, which is really viable immune tissue, put it in the people with HIV, maybe it would restore some immune function. We did the process. We went through taking horse globulin to make us so we wouldn't become immune to the thymus tissue that was not our own tissue. It was foreign tissue. Everything was safe. Everything was fine, except guess what came along during the surgeries, heart therapy. So heart therapy got the main stage, and the research on that whole era of thymus research stopped. The good news was it was safe, and the tissue that was put into my body from a baby was viable tissue. But that's all we know about it.
And what is heart therapy for folks that might not know what that is?
Heart therapy is highly active antiretroviral therapy. It's typically not called that anymore because all the therapies we use now are highly active. But at the time, it was a cocktail of 2 or 3 more drugs that were highly effective.
And just for those who don't know, the thymus is where you make T cells, and it goes away usually after adolescence. So that's why you have a hard time making new T cells, and that's why this idea was tried to see if you could make it like a baby again and make a new immune system.
And can I just add one thing about, I'm so fortunate that I was in a community and in a setting where I could learn all of this stuff that made me want to be a part of it. Normally I wouldn't have ever jumped into a study like that, but I was immersed into all of that activism so heavily that I saw that possibility, and someone invited me to be in the study, probably because I was part of the immune restoration think tank, which was a Martin Delaney's thing, and I signed up. And who knew? Maybe that's why I'm still alive today.
Thank you.
It's really tough act to follow. He's not telling you all of the studies that he was in. So I think there's a couple things. One, there's how when I moved to San Francisco in the late 80s, it was like moving into a war zone, and I became very angry, and my tendency was to self-destruct with anger. So I focused on doing something, and even if it would be a failure, to me it would be more important to try and fail than to not try and fail. And so somehow in our ACT UP in San Francisco, we had a treatment issues committee that really just studied immunology, and we'd read textbooks and things all the time and talk with researchers all the time. But really what we did is we, like yourselves here, is to make research happen. And I think the thing about research, like, I just want to be clear about my own motivation behind it. I never really had the motivation that, like, I'm going to get some stellar benefit from something, because I never thought I'd survive this long. It was for, like, for future folks that like, well, if you're going to answer this question, maybe it'll benefit people down the line, because there's future generations. And so I really kind of took that perspective about it, that it had really nothing to do with me at all. I did do some desperate things, like we were able to make some medications, like DDI, for example, back in the day, and get those on the underground. And I did take part in those before they were approved. I was fortunate to be in a situation to access that, and that is not something everyone can do. And it's simply because I was involved in activism. And I always have this feeling that when I have access to something someone else doesn't, that, sometimes I feel bad about it, because I want everyone to have access to everything. And that's one thing about trials. It doesn't really address social determinants of health, as they say. It doesn't, get you food. It doesn't, what it does is, I guess the simplest way for me to say it is, my perspective is that trials are not treatment. Trials are trials. You might get a benefit, you might not. That said, I was involved in a lot of community advisory boards, including the AIDS clinical trials groups. Somehow I ended up doing neurology research. I'll just leave that, in that area, I'll just leave that alone. We all had our niche, and I was told to do that, so I did it. But I was on IRBs and quite a few things. But the whole point of that is, my whole motivation was to make research happen, and to do whatever it takes to make research happen. Incidentally, in the state of California, if you have a lot of civil disobedience on your record, it doesn't go away.
How often do you think about the occurrence of the natural genetic mutation, given its distribution not matching with where the epidemic is seen globally?
So I began working with Sangamo in 2004, actually, and we did the first patient, and you heard these, Matt's got it, but our first patient was treated in 2009. But the original gene, CCR5, was discovered because of hemophilia, which a lot of people don't know. Hemophilia, is mostly in boys, because it's a sex-linked gene. And when the HIV epidemic happened, initially they didn't have blood tests. So, and the hemophiliacs got blood transfusion all the time. So they got infused IV with live HIV. And I was in Philadelphia Children's Hospital. All the kids with hemophilia got HIV and died. But a few survived, and when they tested them later, they found out they had a natural mutation in the gene called CCR5. And we now know that this occurs in, in both, you have two chromosomes, so on both of your copies can be non-functional, and if you have that, you won't get HIV, even if you get an IV infusion. So, and it only occurs in Northern Europe. We don't know why it didn't occur in Africa or in Asia. And the thought a lot of people have is it happened because of the plague epidemics in Europe, which were caused by rats, and a lot of people died, and it's a really big activation of your immune system, and that may have helped you survive if you happen to have that. So now what we have is, it's a marker of people who won't get HIV. And then now, and it's been used now, can we, the cell and gene therapy approaches, can we introduce that into someone who didn't get born with it? So, 10% of people have one of the copies out, and about 1% or 2% have both of them, and they're completely resistant. But now you can, with cell and gene therapy, you can make that happen to everyone. And that's what the Matts here have gone through.
I love that question. That's a really, really important question, and I do think about it all the time. So back up to the late 80s, early 90s, is what would happen a lot that people probably don't know about is you would have researchers that were scared of you because you show up angry, and sometimes researchers, but usually drug companies, would try to give you a drug that isn't accessible to everybody. And what our group really held to is that we don't want things that aren't accessible to everybody. So on one hand, there's studies, and studies are studies, and not very many people go into studies. I mean, they're always, like, my thing is that they're there to answer a question. But as far as, I guess one way to put it for me is that, activist skills are portable skills. And so, what you do as an activist, you work on a macro level of things. And when, for me, what I did is took that to individual level. So you can take, like, all those skills and use them in hopefully a productive way. But what I don't want is I don't want a treatment that not everyone can get. And I think the researchers here can answer how complicated that is with something like gene editing. But yes, to me, I think about it actually daily.
So actually, by going through the therapy, throughout those processes, you said groups. So what was it like going through with the therapy, and then you come in with, did people know that, were you in groups with people that you knew that were going through the same therapy process as you, and you coming out of that process, how did they feel with you being cured? Did that make sense? Oh, I mean,
well, what's interesting is the way that they had the study. The study I was in, I'll go into detail later, but the way it was set up is that they staggered the folks that were in the study, and so that you didn't meet other people that were in the study, and they kept their confidentiality very well. But the weird thing about, just to digress a little bit about the study that I was in, is when you get your cells, you smell like rancid cream corn. So I could figure out by who's in the office, who else got or who must have just got their cells. And so I only knew a couple people that were in the study site that I was at. So we didn't really, to my knowledge, and Matt might be able to expand this, I don't know all the people. There wasn't really a group, like I don't know those people, but there's a lot of medical reasons why I wish I did, because I think that overall, globally, those of us that were in the study do much better afterwards, because a lot of people were kind of treatment failures all the time, like myself included, quite a bit. But I'm going to pass it to Matt.
I would just add that, when you enter any kind of clinical trial like this, you sign a bunch of documents, right, to sort of sign your life away. Not really, but you want to make sure that you're protecting your confidentiality. So usually you do that in a private setting with a doctor. And so if you're in a study, the only other person you will know that's in your study is just because you happen to know they're in it and you talk to them. They may even be coming to the same clinic, but they may be in another complete study. So you really never know. And in the Sangamo phase 1 study that I was in, I was actually invited to be into that study by Jay Lalazari, who runs a clinical setting in San Francisco, who approached me and said, this is a perfect trial for you at this point in your life. And so that's the only way I knew. And then the other way I knew was after the study was over, people would post stuff on like Facebook, right? So you learn people who are in the study at that point. But that's...
I think that's a really interesting segue from, all the HIPAA rules we have to protect confidentiality came because of HIV. But it's had some negative effects. I mean, we don't know it actually slows down research sometimes. Like if you want to do like what was done for Tim Brown and give a transplant for someone who has leukemia and give him a bone marrow donor who has the CCR5 gene that you want, we can't do that in the United States because you can do it in Europe, but our HIPAA laws don't allow that. We can't go back without it getting consent from everyone. And where it's been broken is what Matt kind of just hinted at. So in my case, I started treating kids with leukemia, and sometimes we had spectacular responses, some not. But the parents made these Facebook groups, and everyone all of a sudden knew who everyone was who was on the trials. So the parents actually learned about the progress faster than the physicians did, because you had to wait to get it published and all that. The Facebook has accelerated research in some ways, and it's unblinded thing. So it's a really interesting thing you brought up, Matt.
So talk about more of your experience going through the clinical research trial. What was your experience in the process like for you personally, emotionally, mentally, physically even? And did anything particularly come up for you that maybe you were taken back by?
Wow, so much. I mean, at the time, I was approached by the investigator of this zinc finger trial. It was a safety study. It was the first. This is right. It's the first gene therapy trial in HIV. Correct? The zinc finger. No, that's our second. Do you want to tell a little bit of the history of that, or? So
there's one thing is gene therapy is putting in a gene, and that's called gene transfer. And the first trials on that were with CAR T cells, actually, and we did that in South San Francisco with a company that's now out of business called Cell Genesis. And so we treated about 40 patients. We actually found it very safe, and that it's actually, those patients still now, 25 years later, still have CAR T cells. So we learned a lot from that. What Matt's talking about is gene editing, which has only been possible in the last few years, where you can actually find the needle in the haystack and take your own genes and change them how you want them to be. So it's not the addition of a gene, but it's just rewriting the genetic code.
And I was looking up at a slide, the first gene therapy, and correct me if I'm wrong, the first gene therapy patient died. Actually, it was a J&J ribozyme trial. Is that right?
So the whole history of gene therapy, is complex.
It's complex.
And it had some initially biotech involvement, and then biotech got out of it because they thought it would never become commercialized, and it became all academic. And then Sangamo came back in with what the Mats have been on, because they had gene editing and technology. But there were from fits and starts, and then there were some toxicities that made, so the initial trials, and they got very highly publicized, and everyone got afraid of it.
Yeah. Thanks, Carl. I think for me, it was my salvage, really. It was the way I was going to stay alive, because I'd run out of treatment options. As I said before, I'd used every drug class that was available, and I kept waiting for the next new drug, and I just knew that wasn't going to last me. My T cells started falling. Fortunately, my viral load was suppressed at the time, but when I had heard this opportunity of an immune-based, like an immune-based therapy, really an immune-based therapy, because it was using my own cells, just manipulating my own DNA, that I might have a chance. So I jumped at it. And I honestly, I talked to a lot of these guys, these scientists about it. Is this going to be safe for me? Do you think this is something I should try? I didn't go at it blindly. I certainly did my research, talked to, people, Martin Delaney, the people that knew their stuff. And they comforted me enough to, you'll probably be okay. And they were probably gambling just by telling me that, right? But it was fine. And the procedure was literally, you may be surprised at hearing this, literally, does everybody know what a bone marrow apheresis or a pheresis procedure is? When they take your blood out of one arm and they separate it in a machine, and they collect, in this instance, the white blood cells, and then they put the red blood cells back into your body So you got all these tubes running out of your body. It's about an hour-long process, but it's a way for them to collect a large amount of cells. And they need those cells in order to manipulate them and to, in fact, they grow them in the laboratory, and then they infuse so they're my own cells. They infuse them back into my body. So there's no really chance of me becoming having a allergic reaction. There's a little chance of that happening. I didn't. I waited about a month. I got a call to show up at the clinic. The product, they called it product, was this little white bag. It looked like mayonnaise almost, in a white IV bag. It was frozen, solid. They had to thaw it out, and they infused it into my arm in 20 minutes. That was the only thing I did for this trial, except then later come back for blood draws, and I had I think it was something like 12 sigmoid colon snips to test my colon to see if the cells had gotten into my colon. And that was not exactly fun, but it was certainly something that I got over, and I did because I knew it was something that hopefully would work for me. And that was it. Basically, year 2 years later, the results of the study for me were not expected, and for almost everyone I think in the trial, their CD4 cells doubled. Now, we were all people who had been on antiviral treatment for years. We're called immunologic non-responders. We were people who had suppressed our virus with the available drugs, but our T cells never rose like a lot of people's T cells rise when they get on effective treatment. But most of us had started late, or for whatever reason, we didn't have that rise in T cells. So we entered the study. As a result of this one infusion of these manipulated cells, we all had a doubling of our T cells. It's been 9 years now, and my T cells have remained at that level. And and remember, I'm still on treatment, antiviral treatment. But I shouldn't have gotten a doubling of T cells just because I was in this trial. Does everybody get that? Did I explain that good enough?
I mean, what Matt described is called a first-in-human trial. You don't know what's going to happen. The doctors don't know. The patients don't know. We had results in mice that where we gave HIV to mice, and it looked promising there. But what we saw in humans was not expected. I mean, I had one patient whose CD4 count went to 1,200, and it was above normal. So all of a sudden, he's above normal, and he'd been in the basement before, and so. The patients really, I think, have benefited from that, and that's not what we were looking for, but it happened, and it was reproducible. And so now, there's a lot of new doors to look at because of that unexpected finding.
And can I just add one thing? When they presented the data for this study, I happened to be in the audience. I think it was here. It may have been in Boston or here. But I was sitting in the audience, and they were presenting the data. And I knew my patient number, and I saw it on the graph. And literally, when they showed the T cell slide with everybody's T cells doubling, the audience, scientists, gasped. You never hear that. You rarely hear that at conferences, but you could hear an audible gasp because this had never been seen in a gene editing study before. It was quite an amazing time. And for me, I was like, "There's my number. It's going up." It was great. It was great.
So, when protease inhibitors came out, for me, I kind of got this, oh shit, I'm going to have to get a career moment. And moved and decided that I need to go back to school I'd already been doing like harm reduction work and stuff, and I decided to go back to school so I could get paid like the other people that were doing that job. And I stepped away from activism and just kind of followed it. But my experience on antiretroviral treatment is one where I frequently became resistant to drugs. Up until the drug centrist came out, I would usually have around 300 T cells as a baseline. and I would have a 20,000 copy viral load as a baseline. And for some reason, by the time, like Matt, but not as drastic or dramatic, by the time that Timothy Brown was cured, if I can use that word, I was left to just 2 protease inhibitors and an entry inhibitor called centrist, and that was the first drug that got my viral load down to 0. What piqued my interest, I'd kind of known about the CCR5 for a long time, since the early 90s, and what piqued my interest was when Timothy Brown was cured, and I started to really think about, like, well, you know, someone's been cured, like, you know, make this research happen, you know, like maybe you can replicate this in different ways. And what I'm trying to say is that I got this kind of tinge of optimism and hope that I just kind of been gliding along, like okay, I'm on meds, I'm working and stuff, and activism changed, and I don't really need to do that as much. And so I've always been in touch with Matt. We go back way back, like I said, and I knew about his study and when that happened, and I paid very close attention to it. I'm someone who's like I go down a rabbit hole with research, like I just have to understand it, and until so I'll just bat my eyes and ask somebody to explain it to me until I get it. So when his study came out, I could see the changes. Like I know him, without him telling me, I could see the changes. When the study I was in is what's called a phase 1/2 study, and what they were doing is, when you get your altered cells, you have to, you have to give something to suppress the immune system. So what they were trying to do was to give a dose of chemotherapy, and they were trying to, what they were doing was called dose escalating, to see like how much can we give that people can tolerate that affect your immune system enough so that you'll accept these cells. And in transplant, for example, all the time, people often don't get the kind of suppression that they need. So it's a well-known thing. And so I think I was probably the 6th person that got cells. I had already been through chemotherapy before. I had lymphoma. I had like stage 3 lymphoma, so I did what's called a CHOP with Rituxan. And so what I'm trying to say is that the chemotherapy they gave me was nothing compared to that. It was like, yeah, no problem, I feel kind of crappy for a day. So I tolerated it really well. I might have been a bit of an anomaly because I had 6 bags of that they grew out. And when they grow out your cells, they put it in liquid nitrogen, and I had a bag that broke. But so what I'm saying is, it took me about an hour to get my cells. So I didn't know back then that was a large number of cells. And I don't know if that had anything to do with why I'm in the situation I'm in right now. I should really say though, like I went into the study not thinking that, oh, I'm going to be cured like Timothy Brown. That wasn't my motivation. My motivation was that if you're going to cure HIV, this is one of the ways. Genetics is perhaps one of the ways to do it because it's already been done, and maybe that's the answer. And I'm willing to participate in something like that. I think it's a very important question to answer, even if it fails, or even if it just gives you more information to go on for perhaps combination therapies of other kinds. It was planned that I would go off meds. You stay on your meds for 12 weeks. And then when I stopped meds, it was really interesting. I think going back to something I said earlier today, like for me, when I was taking HIV meds, I had this daily reminder of this whole disease of society. I know I keep saying that, but it's actually true that I take my meds and I would think about like, oh yeah, you're taking all your dead friends and all that kind of stuff. And it was just something that really stuck to my mind. And so that's, like all of a sudden you're not like, oh, I'm just not doing this. And I didn't physically feel a lot different, to be honest about it, but it's interesting to just roll through that experience. I tolerated the treatment really well, and very fortunate. And my kind of thing now is that I know I still have virus, and sometimes I think it's not a if, it's a when. Like someday this might take off, and I'll probably get back on meds, and I'll just ride it out as long as I can. I actually went through chemotherapy and radiation again in 2016 while I was in the study. And much to my doctor's dismay, I did not go back on meds. I said, well, let's see some labs. And what happened is that ever since that point, my viral load was actually lower than it was before. And they think it's maybe because of the radiation, but it stayed at 100 or below. T-cell rebound, it's kind of the same. I kind of bounced between 300 and 400. I didn't have a big, huge, increase in T cells, except for right away, which were 24,000. Really? Yeah, my first.
So my question would be for Dr. June. What is one of the surprising findings that you see in the field of cell and gene therapy? And then I want to refer to the documents that are at the table in the back. So as it was alluded to at the beginning of the meeting today, is that the Defeat HIV Community Advisory Board has conducted social science research on topics of cell and gene therapy or other cure approaches to HIV. And I want to ask a question from one of the focus groups on acceptability of cell and gene therapy. So on top of the surprising findings of the field of cell and gene therapy, one of the questions that was raised in the focus groups was, how are adverse effects taken care of in these kinds of trials?
So Manuel, the first thing is, the biggest surprise to me now is that cell and gene therapy has become accepted as, it's a new pillar in medical care. So it will cure diseases that were previously uncurable. So that in 2017, the first gene therapies were approved, and it's for leukemia. Very soon, an uncurable disease is sickle cell anemia. That's going to be curable with gene-modified stem cells. It's a huge breakthrough. And so I think we should all, this science is amazing what's happening, but I want to caution you, it's going to take time till this goes out. I think it's 10 years really for HIV till we have something that's FDA approved. It goes faster in cancer, because you can take more risks there. But what cancer has done is pave the way for a non-fatal disease like HIV. So I am confident it's going to happen because we have the scientific tools, from gene editing and others making new kinds of cells and so on. So it's really a very amazing time now, and it's a question of how long it will take. Now the side effects, we don't know. Everyone's followed with, right now, if you get a gene-modified cell like the two Matts did, you're supposed to be followed for 15 years, and the FDA is doing that. It's a very good policy, so we know if there's long-term effects. And I mentioned, the first patients we treated with CAR T cells with HIV in 1996, we treated 40 of them, a lot of them in San Francisco and some at Harvard. None of them have had side effects, and they've all been followed, and they still have the CAR T cells. So we think it's very safe, but each of these things needs to be tested, and unfortunately, in a disease that's not cancer, it takes longer. You have to be very careful. So we need to be, aware it's going to take longer than we wish it would. But I am really confident it's going to happen in this next decade.
It's your shoulders that those of us living with AIDS stand on, and we owe you such a deep debt of gratitude. So thank you.
What's it like to go off therapy? With U equals U now and all the emphasis on untransmittable, undetectable, what was it like to go off therapy and not be on pills? You talked about the upside of not being reminded, but was there a concern about that? And like you said, your viral load may rebound at any time, so you don't know when that might happen.
So I think part of what I hear in that question is do I have some level of fear about whether my virus is going to take off or something. I actually am pretty confident that there's so many different classes of drugs that come out that that I really don't have that fear. And the other part about that is I'm going to accept whatever comes my way anyways. Because in other words, like, why shouldn't something bad happen? You know, things like that happen in life all the time. You know, and why would I be an exception to it? But it is interesting, again, on the psychology of not taking medications is actually really, really interesting. It's a routine. It's part of life. It's part of who I was and who I am, I should say. And I never really had an adherence problem. In other words, you know, I'm not perfect. I don't know anyone who is with adherence. You know, I'm sure there's people out there that that are. But you know, it was just it was just like you get up in the morning, you take your meds, you go to, you know, and you take them, you know, in the middle of the day, and then you take them later, and it was just became routine. So that routine changed, but I'm also on other meds. I still take those. You know, they're not HIV meds, but you know, there's other things like I'll spare you that, but yeah. So I hope that answered your question. I don't have a big, robust answer to that question, though. I think, in other words, everyone would probably have a slightly different experience.
What about opportunistic infections like Kaposi sarcoma and the disruption associated with antiretrovirals?
I think Dr. Jerome here knows more about that than any of us up here. You know, Kaposi sarcoma is caused by a herpes virus, and so there are therapies directly aimed at that, and there are therapies directly aimed at improving your immune system so that it can control. So I think, you know, and just like, I mean, you heard Matt here say he got a lymphoma, and there are HIV-associated lymphomas, and there are specific therapies, but one of the best things is restoring the immune system, and then the you know, the chance of getting secondary cancers like opportunistic infections goes down.
So one of the drugs I take is a gram of valacyclovir, which I was on anyways because I have other viruses like herpes. And don't like to say that publicly. I don't know why. But so that might be one way that it's addressed. I don't know. I actually never got KS even before antiretrovirals came out. But one of my best friends died of it. He got it in his lungs, and he went really fast.
Jeff, I think one of the things I think about is, I mean, this is really a whole new era. I mean, if we get to this point where we can get people walking around like Matt, who can be what we call, the term is functionally cured, right? So you still have HIV in your body, but you're probably going to be so low that you're not going to be able to transmit.